Good afternoon to the class and welcome to good afternoon. Yeah, and welcome to practice problem session week 6. In this session, we'll be solving some problems corresponding to lecture week 6, where Professor Subramaniam, in the course of Introduction to Developmental Biology, is teaching about cell-cell communication. And we will have questions related to that and hope this problem solving session helps you clear your concepts and help you in solving the actual questions in the quizzes and in your exams. So we'll start shortly at 305. Wait for 5 more minutes for more students to join, and I'll keep admitting students who are trying to request to join this link, and I'll start sharing my screen at 305. And the general instructions to all students who have attended the previous classes, as well as the new students who have joined. So you please turn off your video chats and mic, and it will be a two-way session in which we'll pose some questions and we'll try to find out the correct answers of those questions. You can post your answers in the chat box, or if you want to talk to me or you want to have a discussion, you can raise your hands and I'll let you speak. So for smooth conduction of this session, you try to communicate maximally to the chat box, and we'll begin soon at 305. Thank you. All right, since it is already 305, we will start our session. And meanwhile, I'll keep admitting people who are trying to join this call. Let me share my presentation, and you guys can confirm with me in the chat box whether you're able to see the slides and if you're able to hear my voice clearly. Let me share my presentation just in a while. Ajay, are you able to hear me now? I think you have reconnected, and also there was another student, Rahul. He is also reconnecting. Okay, great. Please let me know if you can see my slides and can you see the slides changing? Are you able to see my slides? And the slides are changing. I think it's taking some time, but might be some network issues. Ajay, are you able to see the slides? Let me know with a yes or no in the chat box. Great, great. So let's start this problem solving session for NPTEL Introduction to Developmental Biology. And this course is run by Professor Subramaniam from IIT Madras. And what we'll be doing is we'll be solving practice assignment for week 6, where Professor Subramaniam is teaching about cell-cell communication. And my name is Ashutosh Bera. I'm a PhD student at IIT Kanpur. So the general procedure which we follow in these practice problem solving sessions, we take some exemplar questions or some previous year solved questions from the corresponding lecture series, and we try to solve it, which in turn will clear our understanding and concepts and then help us to solve the actual problem sets that we encounter in the final exam as well as in the quizzes. And it will be a two-way session, so I expect the students to participate and try to guess the correct answer or try to find out the correct answer, and then we'll slowly move on to the explanation behind why the correct answer is the correct answer. Moving on to the first question: in what order do these events occur in the hedgehog pathway? This is the question. In what order do these events occur in the hedgehog pathway? And you have to choose the right option. Second slide is not visible. Okay, okay, let me check again. Now I hope you are able to see the next slide. There is question number 1. Is it visible now, Ajay? Yes, great, great. In what order do these events occur in the hedgehog pathway? Is the question. Choose the right option. So there are a series of events that are happening in the hedgehog pathway, and what we have to do is determine the correct order of events. First statement: hedgehog binds to the patched protein. Second statement: hedgehog activates smoothened. Third is patched stops inhibiting smoothened. Patched inhibits smoothened. Smoothened activates pKa and slim. Smoothened inhibits pKa and slim. Smoothened phosphorylates cos2 and Ci is released. Ci enters the nucleus, recruits CBP and activates transcription of hedgehog responsive genes. Ci is cleaved, it enters nucleus, recruits CBP and activates transcription of hedgehog responsive genes. So out of all these statements, we have to find out which are the correct statements, and we also have to see what is the correct order of events happening in the hedgehog pathway. And there are many options downside the question. I hope you can see them. There are many options here. Statement 1 followed by 3, 6 and 7 simultaneous with 8. Next option is statement 1 followed by 3, 6 and 7 and last is 9. Similarly, we have 1, 4, 6, 7, 8, 1, 4, 5, 7, 8, 1, 3, 6, 8, 2, 5, 9, 1, 2, 3, 6, 7 and 8. Now which of these options do you think is the correct answer? Let me know in the chat box. Take a minute to read the question, go through all the statements, and whichever you think is the correct order of events happening in the hedgehog pathway will be the right answer. I think Ajay, have you answered this question? You have mentioned 1 in the comment box. Does it mean the first option you're talking about? Yes, sir, first option. Okay, okay, great. Now let me reveal the correct answer to this question. Ajay has answered very correctly. First option is the correct order of events happening in the hedgehog pathway. So the first event that occurs in the hedgehog pathway is hedgehog binds to the patched protein. So patched is the receptor for hedgehog. Number 3 statement, patched stops inhibiting smoothened. So smoothened is another conjugate protein which is inhibited by patched. Then what happens? Smoothened inhibits pKa and slim. Smoothened also phosphorylates cos2 and Ci is released. These are two simultaneous events followed by the final event where Ci cubitus interruptus enters nucleus, recruits CBP, activates transcription of hedgehog responsive genes. So this is the correct order of hedgehog pathway and Ajay has answered correctly. Let us have a simplified look about the signal transduction of hedgehog pathway. So here is the schematic on which our answer was based. Hedgehog inhibits patched. Patched was inhibiting smoothened generally, but now since patched is inhibited, smoothened is free. Smoothened activates Ci protein made activator, which goes inside the nucleus and turns on transcription of the target genes. So what happened in normal states when there is no hedgehog ligand? The patched protein is the receptor for hedgehog ligand, inhibits smoothened. Smoothened is another membranous protein. Now when this smoothened protein is inhibited, there are two other players which are active, protein kinase A and SLIM. So what does these do? Protein kinase A and SLIM, pKa and SLIM, cleaves the Ci or cubitus interruptus, which is the mediator molecule of hedgehog signaling. Now the cleaved Ci cleaves and phosphorylates Ci. Cleaved Ci can go and act as a repressor on responsive genes. Now when there is presence of hedgehog ligand, it binds to the patched protein. Upon binding to the patch protein, there is a conformational change in which the patched protein or the receptor is no longer able to inhibit smoothened. Now smoothened becomes activated and smoothened inhibits two of these players, pKa and Slim. Thereby, now pK and Slim would not be able to perform their function, which was earlier to cleave and phosphorylate Ci and act as a repressor. So pK and Slim will not be able to perform their function, and cos2 will be phosphorylated, Ci will be released, and whole Ci, not cleaved Ci, will act as an activator mark, recruiting CBP, and transcription of hedgehog responsive genes can follow. So this is a rough schematic of how the hedgehog pathway works. The hedgehog pathway is a classic example of negative regulation, wherein signaling pathways that are generally inhibited but have their inhibition removed upon activation to trigger specific cellular responses. So the ground state of this signaling pathway is negative, means it is turned off, in the way that patched receptor is always inhibiting the smoothened protein. So the ground state of the signaling pathway is inhibition. Now what happens when an activating ligand comes up? The pathways that are generally inhibited have their inhibition removed upon activation to trigger specific cellular responses. So the hedgehog ligand will remove the inhibition of inhibition of smoothened by patched receptor, and that's how it will proceed to a activating specific cellular response. These pathways are tightly regulated to ensure that cellular processes occur only when needed. So why do we have negative regulation? We have such pathways which are tightly regulated to ensure that cellular processes only occur when needed. Means it cannot be activated, cannot be on in the ground state. Only when it's needed, it can be on. And another example of such signaling is Wnt beta-catenin signaling, in which the ground state of Wnt signaling is off due to degradation of beta-catenin. Now once the Wnt ligands comes into picture, the beta-catenin degradation complex is inhibited, and now beta-catenin can go inside the nucleus and turn on specific transcription, Wnt responsive transcription. And SHH-CI signaling. SHH is sonic hedgehog, one of the prime ligands of hedgehog family, and CI is cubitus interruptus, the mediator molecule which translocates inside the nucleus and causes transcription of downstream regulator genes. These are an example of classic negative regulation, and we also studied the hedgehog pathway of signal transduction, and the order of events happening in the hedgehog pathway. I hope it's clear to the audience. Just let me know with a yes or no in the chat box. We can quickly move on to the next question. Great, great, Ajay. So moving on to the next question. Which of these are false? So there is a list of statements and you have to find out the wrong statement. Which of these are false? Statement 1. In Jack start signaling pathway, start acts as both the transducer and the activator. Statement 2. Prolactin acts as a ligand for activation of casein gene through the JAK-STAT pathway. Statement number 3. If there is a mutation in the N-terminal of hedgehog protein that prevents cholesterol attachment, the hedgehog pathway will be constitutively activated. And statement 4. If there are problems in synthesizing cholesterol, it may affect the activation of genes that utilize the hedgehog pathway. So there are a certain set of statements, two of them belonging to the JAK-STAT signaling pathway and two of them to the hedgehog pathway. So now you have to identify which of these statement is a false statement. I think Ajay has already answered option 3. Ajay is saying option 3. Now let me reveal the correct answer. Ajay is very correct. Option 3 is the correct answer. Option 3 is a false statement. If there is a mutation in the N-terminal of hedgehog protein that prevents cholesterol attachment, the hedgehog pathway will be constitutively activated is a false statement. Rather than the hedgehog pathway will be constitutively deactivated. And we will all understand about all these statements about JAK-STAT pathway as well as the hedgehog pathway in the upcoming slides. So if you see, this is an example of prolactin hormone signaling through the JAK-STAT signal transduction pathway. So this diagram will answer the first two statements where we talk about JAK-STAT signaling and prolactin ligand. So prolactin is the ligand here. Prolactin ligands causes dimerization of activated receptors, and these receptors are conjugated with protein Janus activated kinase JAK, JAK2 here in this model. So when these receptors dimerize due to the presence of the ligand, these dimerization will cause phosphorylation. So JAK2 will cause autophosphorylation, and each of them will phosphorylate themselves and cross phosphorylate. This phosphorylation will trigger another protein which is cytoplasmic called the STAT protein. Now STAT protein stands for both. STAT protein is a protein which can act both as a transducer as well as the activator. So the transducer, it means that whatever the external signal is received from the plasma membrane is transduced by the STAT protein. So this signaling pathway do not have any other adapter or scaffolding proteins. The STAT protein is the transducer protein. Also, active STAT now, when phosphorylated by activated JAK proteins, phosphorylated STAT can form dimers, STAT5 dimers, as in this case, and this dimerized STAT can also act as an activator where it can translocate inside the nucleus and turn on the transcription of its target genes. So STAT here is both a signal transducer as well as activator. So STAT full form stands for signal transducer and activator. STAT transduces the external signal via phosphorylation, and phosphorylated STAT can dimerize and form an activator complex which can translocate inside the nucleus and turn on the downstream transcription of casein gene promoter, as in case of prolactin hormone. So this answers our first two statements. These two statements are correct. JAK-STAT pathway, the STAT is both the transducer and activator, and prolactin turns on the casein gene promoter or casein responsive genes. While on the other hand, in the first question we saw about the hedgehog signal transduction pathway. So as we know, hedgehog proteins are initially synthesized as precursor molecules that undergo several modifications before becoming active signaling molecules. So if we talk about the ligand here, many ligands of different signaling pathways modifications undergo modifications which can be then recognized by the receptor, right? And one of these modifications in case of hedgehog proteins involves attachment of a cholesterol to the N terminus of the hedgehog protein. That's what I have mentioned here. So the N terminus of hedgehog protein will have this cholesterol modification. Now cholesterol attached hedgehog protein can be recognized by its receptor, the patched receptor, and it can activate the patched receptor. Cholesterol modification enhances the affinity of hedgehog ligands for their receptor patched and allows them to more effectively inhibit patch. So cholesterol modified hedgehog protein can more effectively inhibit patched and thereby turn on hedgehog signaling. This inhibition of patch is a crucial step in hedgehog signaling because it de-represses the activity of another receptor smoothen, allowing smoothen to activate downstream signaling events. Therefore, the third statement, which is the correct answer, is a false statement because cholesterol absence would render the hedgehog signaling pathway constitutively inactive. So, if you do not have the N-terminus attachment of cholesterol to the hedgehog protein, in fact, we will have the hedgehog signaling pathway constitutively inactive. It will always be turned off because now the ligand cannot recognize the patched receptor, and the patched receptor will always be inhibiting the smoothened receptor, thereby uh, may, thereby inhibiting the hedgehog signaling pathway, constitutively inactive. So that is the correct answer to this question. I hope it is clear to the students. Yes. Oh, okay. Ajay has a question. Ajay is asking what releases prolactin other than breastfeeding. I think this is the question. Okay, so prolactin is a hormone, as you all know. Prolactin hormone is generally secreted in pregnant mothers for breastfeeding, but also there are reports where there is another pathway which can affect the levels of prolactin is a dopamine pathway. So dopamine is a neurotransmitter that is released as a reward pathway. So if you do something, your brain rewards you by secreting dopamine. So such neurons are called dopaminergic neurons, and they have a particular neurotransmitter known as dopamine, which activates the reward pathway. So now the person will do complex activities to gain that reward. So if there is any medication affecting the dopamine reward pathway, it can also affect indirectly the prolactin levels. So there are certain antipsychotic medications and certain high blood pressure medications which can affect the secretion of prolactin, because prolactin is uh, is made in the pituitary. Okay, yeah, let's move on to the next question. So our next question is about wind signaling. Wind signaling pathway is not involved in which of the following? Wind signaling pathway is not involved in which of the following? Induction of ventral cells to form cartilage, induction of dorsal cells to form muscle, development of urogenital system, limb and midbrain development, proliferation of stem cells. Now this is a direct question. If you guys know about the properties of wind signaling and where do you find it the most active, you might be able to answer this question. Induction of ventral cells to form cartilage, induction of dorsal cells to form muscle, development of the urogenital system, limb and midbrain development, proliferation of stem cells. So Ajay has already answered option 1. Let us check if option 1 is the correct answer or not. Option 1 is the correct answer. Induction of ventral cells to form cartilage. Wind signaling is not involved in induction of ventral cells to form cartilage. Whereas how we will see that wind proteins are critical in establishing variety of functions which include polarity of insect and vertebrate limbs. The polarity of insect, so wind, as the name suggests, wind stands for wingless and integration site 1. It is a portmanteau term for a protein which was first discovered in Drosophila and in mouse. So in Drosophila, as the name suggests, after the phenotype wingless. In Drosophila, when the Wnt protein was mutated, a wingless phenotype was observed, and Wnt was discovered as a segment polarity gene. So in insects, it is a segment polarity gene. It establishes the polarity within segments of the Drosophila body and also in vertebrate limbs. Wnt is also a pro-proliferation marker. Remember, in any cases, in various cases in the body, you see Wnt is always involved in cancerous growth, in causing cell proliferation. So Wnt is always a pro-proliferative marker, whereas on the other hand, we have BMP in general. So many cases you might find Wnt also plays a role in differentiation because it is such an important ligand. It has roles in many different processes, but generally Wnt and BMP antagonize each other. BMP is pro-differential while Wnt is pro-proliferal. So Wnt is also responsible for promoting proliferation in stem cells, in regulating cell fates along axis of various tissues. So Wnt is also a fate determinant, and development of mammalian urogenital system. The urogenital system consisting of ureter, kidney, and the nephrons, the bladders, everything is dependent on Wnt signaling, very important, and guiding the migration of mesenchymal cells and pathfinding of axons. So Wnt also plays a role in mesenchymal cell migration and axon pathfinding. So these are variety of functions that Wnt plays a role. And if you see that induction of ventral cells to form cartilage is done by another ligand molecule, very important signaling pathway called SHH, sonic hedgehog, from the hedgehog signal transduction pathway. So here is an example of mouse embryo at day E9.5, where we see cartilage initiation site marked by the expression of SHH. Similarly, tooth initiation site as well as nasal prominences. So if you see, these nasal prominences are ventral structures. If you divide our body into dorsal and ventral parts, the nasal prominences or the nose is a ventral structure while the spinal cord is a dorsal structure. So SHH is majorly responsible for ventral induced structures such as cartilage, and SHH is also found in zone of ZPA or zone of polarizing activity in the AER in the limb bud. So this is a limb bud or something which is a primordia of a limb from where the limb of an organism protrudes. So there we have different signaling centers of which SHH form the dorsal, the posterior and ventral side of the limb bud, and SHH is responsible for cartilage determination. So in a summary, Wnt is primarily dorsally active and induces dorsal structures, while SHS is ventrally active and induces cartilage from ventral tissues. Now if we see in the context of brain development, as we had an option about midbrain development and forebrain development as well as the limb development, you see here Wnt BMP source lies on the dorsal side of the cortex, whereas SSH source lies on the ventral side of the cortex. So most probably Wnt and SHS are marking the boundaries of the regions that are going to form the dorsal and ventral structures. SHH or sonic hedgehog expression marks the ventral structures while Wnt expression marks the dorsal structures. Similarly here we have dorsal and ventral side where we have Wnt 7a expression enhancing the SHH expression in the ventral side while Wnt 7a is present in the dorsal side. And this is a cross section through a limb bud. So here, as the students have correctly answered, induction of ventral cells to a cartilaginous fate is not a work of Wnt signaling rather than hedgehog signaling. Wnt signaling is involved in dorsal induction as well as urogenital system, limb and midbrain development as we see here. and stem cell proliferation. I hope the explanation is clear. We learnt about a little bit about expression patterns in different tissues of Wnt and SHH, as well as the fates that they determine. Great. We move on to the fourth question. The next question now: which of the following are true with respect to the TGF beta pathway? So this is a question about TGF beta pathway. There are many statements and we have to pick out what are the true statements. The receptors dimerize when the ligand binds to them. Ligands can only bind to receptors that are already dimerized. Dimerization of receptors causes their phosphorylation and activation. Activated receptors phosphorylate and inhibit SMAD. Phosphorylated SMAD enters the nucleus and regulates gene transcription. BMP, activin, TGF beta are some of the ligands for TGF beta pathway. So let me tell you that there can be multiple options which can be true, which can be correct. So you have to go through all the statements and put forward your best possible answer. Statement 1: the receptors dimerize when the ligand binds to them. Ligands can only bind to receptors that are already dimerized. Dimerization of the receptors causes their phosphorylation and activation, and activated receptors phosphorylate and inhibit SMAD. Phosphorylated SMAD enters the nucleus and regulates gene transcription. BMP, activin, and TGF beta are some of the ligands of TGF beta pathway. So these are some of the statements. Take a minute to go through all the statements and put forward your correct answers. I see there are already some answers. Ajay has answered statement 1, 3, 5, 6. Okay, let's have a look at the correct answer. Ajay is answering 1, 3, 5, 6. The first statement that is true with respect to TGF beta pathway is the first statement. Receptors dimerize when the ligands bind to them. Receptors do not dimerize without the ligand. Statement number 3 is the second true statement. Dimerization of receptors causes their phosphorylation and activation. So once the receptors are dimerized, this event causes their phosphorylation and activation. BMP, activin, TGF beta are some of the ligands for the TGF beta pathway. Statement 6 is also correct. And phosphorylated SMAD enters the nucleus and regulates gene transcription is also correct. So statement 1, 3, 5 and 6 are actually really correct. Congratulations, Ajay has answered very correctly. Very good. Now let us have a look at the TGF beta family molecules of signal transduction. So what we have here in a general TGF beta family of ligands, we have a serine threonine kinase domain receptor type 2 and a GS box receptor type 1. So we have two different types of receptors, and these receptors homodimerize and form a tetramer, heterotetramer of receptor type 2 and type 1 in presence of TGF beta ligands, which can bring both these type of receptors in close proximity. Now when the receptors are in close proximity, the receptors get phosphorylated, and phosphorylation activates the receptors, which then activates the mediator proteins called SMAD. SMADs are the mediator proteins, and different families of TGF beta differ from the kind of SMAD they rely on for signal transduction. So when we have ligands like activin, nodal or TGF beta, we have the activation of SMAD 2, 3 activation in the form of phosphorylation. So the phosphorylated intracellular domains of the type 1 and type 2 receptor can phosphorylate the SMAD 2,3, and then SMAD 2,3 can recruit SMAD 4, which is also called the co-SMAD, which will assist the translocation of SMAD 2,3 along with SMAD 4, form a complex, and this complex can translocate inside the nucleus and turn on gene transcription. Whereas another family of TGF-beta ligands, the BMP ligand, bone morphogenetic protein ligands, they activate a different set of SMADs called SMAD 158. So phosphorylation of the intracellular domains of the receptors of BMP ligand can activate SMAD15. So phosphorylated SMAD15 is an active readout, is a readout of active BMP signaling. Now phosphorylated SMAD159 in conjugation with the help of co-SMAD SMAD4 can translocate inside the nucleus and turn on the gene transcription. So this is how a general signal transduction happens in a TGF-beta like ligands and superfamily, where we have utilization of different kind of SMADs which differentiate among a path, the among different ligands of the same family. If we detect SMAD 2,3 phosphorylated, we can say that activin signaling is much more active. If we detect SMAD 159 phosphorylated, we can say that BMP pathway is much more active. So ligand binding and receptor activation, this includes some of the statements that we encountered in the earlier question. The TGF-beta family of ligands include TGF-beta 1, beta 2, beta 3. They are secretory proteins and binds to cell surface receptors to initiate signaling. The primary TGF-beta receptor complex consists of two types of transmembrane serine threonine kinase receptor. So serine threonine kinase, it means it can get phosphorylated on the serine and threonine residues, which are OH group containing residues. So we can have phosphorylations on primarily three amino acids which have OH group for creating the phosphoester bond, which are serine, threonine and tyrosine, which have a OH group. TGF-beta type 2 receptors are the serine threonine kinase receptor, and TGF-beta type 1 receptor are the GS receptor. The ligand initially binds to TBR2, so the ligand will first recognize TBR2, inducing a conformational change that allows TBR2 to recruit and phosphorylate TBR1. So once the ligand is bound to TBR2, it will recruit TBR1 and phosphorylate the intracellular domains. Then what happens is the formation of SMAD complexes. Phosphorylated SMAD2,3 proteins form heteromeric complexes with common SMAD, co-SMAD, which is SMAD4. This complex formation is critical for nuclear translocation and transcriptional regulation. So this complex of co-SMAD along with the mediator SMAD, the SMAD2,3 or in case of BMP SMAD159 will go inside the nucleus and perform transcriptional regulation. I guess the audience is now clear about TGF beta family signaling. If yes or no, just let me know in the chat box. Okay, let's move on to the next question. Question number 5, which is a okay, so too much echo of your voice. Okay, I'll try something else. Wait. I think it will be clearer now. I have just closed in with my laptop so that my voice is clear and there is lesser echo. Which of these is false is the fifth question that we are going to solve. Human embryos with defects in apoptosis pathway would be lethal. Mutations in genes involved in the apoptosis pathway can lead to cancer. In notch signaling, the signal can come from cells that are far away from the receptor cell. And the fourth statement, notch signaling is involved in the development of vulva in C. elegans, nervous system in vertebrates, and determination of cells that will become the optic neurons. So similar kind of true false question where we have two statements regarding the apoptosis pathway and two questions regarding another important signaling pathway, the notch signaling. Now, which of these do you think is the correct option? I think Ajay has already answered. Third option. Okay. So Ajay says in notch signaling, the signal can come from cells that are far away from the receptor cell, which is false. Let me reveal what is the correct answer to this question. And Ajay is not wrong. This is the false statement, which is the correct answer to this question. In notch signaling, the signal cannot come from cells that are too far away from the receptor cell. Notch signaling follows a specific kind of signaling called juxtacrine signaling. And in juxtacrine signaling, the ligands and the receptor are from the adjacent cells. So we'll study a bit about notch signaling and juxtacrine signaling and also about other statements of apoptosis. So if you see here, I have a simple schematic depicting the roles apoptosis plays in development as well as in cancerous growth. So in development, apoptosis is required for embryonic development. Many transient structures form which later undergo apoptosis to sculpt the organism. Apoptosis is very important for development of the animal because many transient structures appear in the embryo which are not present in the adult. And the function of these structures is to act as a scaffold or a transient signaling center which can later undergo cell death and sculpt the organisms. Simplest example would be the interdigital mesenchyme which we have when we are forming our hands and the digits. Morphogenetic apoptosis acting as a stone sculptor, elimination of material by apoptosis or programmed cell death, new shape revealed. So when we begin our hand formation doesn't start by five elements or five fingers. It starts as a webbed whole bud called the limb bud, and in the limb bud, as the development progresses, there are five distinct regions which form our fingers, and the tissue in between the fingers is called interdigital mesenchyme. This interdigital mesenchyme eventually undergoes apoptosis to give the final form of five fingers which we have in the adult hand. So apoptosis is very necessary in embryonic development to sculpt the organism. So this is a kind of a sculptor with a chisel and a hammer sculpting different statues. Also on the right hand side, a simplistic diagram shows mutation in apoptotic pathways dispose cells towards cancerous growth. So our body has lakhs and lakhs of cells, and each cell undergoes mitosis and germ cells undergo meiosis many times during our development as well as in the adult life. Some cells undergo regular division, some cells are postmitotic, they do not undergo division, and our cell division, in which the cell is stressed or the replication is also erroneous, can garner some DNA damage. Also, due to some chemicals, radiations, and due to environmental factors, many factors can cause DNA damage, DNA breakage in a cell. Now, if there is apoptotic pathway, the apoptotic pathway can fix. Okay, okay, Ajay. Yeah, let me rejoin, and I'll try it again. Just wait a minute. Just a minute, Ajay.
I just hope that it becomes clearer. Are you able to hear my voice now? Is it breaking? Please let me know, then I can continue. Hello? Now, okay, okay. Yeah, let us proceed for some time and let us see if my voice is audible to you. So, as I was talking again, I'll repeat this explanation again. Apoptosis is required for embryonic development. It is very important process which acts as a sculptor in sculpting our organs and body plans. So apoptosis of transient tissues and structures is necessary for the final form of the organ that is getting sculpted. And the simplest example that we have here is the formation of hand, in which the interdigital mesenchyme, these red tissue, we have webbed feet while we are in embryonic stages, and these tissues in between the fingers act as signaling centers in patterning the fingers, and after a certain development stage, these tissue have to undergo apoptosis to give rise to a final interdigited our final hand structure, where we have gaps between the fingers. Ajay, want to speak up? Yes, please speak up. Ajay, you can turn on your mic and talk to me. Okay, now the voice is less than the noise.
Is it clear now, Ajay? I just have removed my headphone device. I'm just directly talking. Just give the question, sir. I will try to. I'm just directly speaking into my laptop now. I have removed all the headphones and ear devices, so I think it will be clearer. But I think some instrument fault is there or some connectivity. Yes, my voice is clear to you. Na? Yes, yes, yes. I can hear you clearly. I think it's the network issue that is breaking my voice in between. Maybe I have some connection issue, but sir, if it have been network then slide presentation, my voice would have also been breaking. Your yeah voice comes clearer for first 2 3 sentences, and then it becomes like temper somebody is banging the bell slowly. Okay, okay. I'll try to be more clear. Let's okay, sir. We can end the session. We can have it for next session. Remaining question. Yes, yes, yes. Not problem for Yeah, maybe today I have a faulty speaker or something, right? So let's not stall our time here and move ahead. So as we were saying that mutations in apoptotic pathway can dispose a cell towards cancerous growth because apoptosis is an important pathway which recognizes these DNA damages and any cell under stress and can remove that cell peacefully in a programmed cell death manner. And when we have loss of apoptosis, these cells could lead to cancer, as is seen in many cancers. There are mutations in genes involved in the apoptosis pathway. So apoptosis is really necessary. Whereas Notch signaling, which was the correct answer for this question, Notch signaling is an example of juxtacrine interaction signaling, which I was saying earlier. The proteins from the inducing cell interact with the receptor proteins of adjacent responding cells without diffusing from the cell producing it. So it is not a diffusible or secretory ligand receptor interaction, while it is a juxtacrine or imposition. Cells adjacent to each other interact with each other with the ligand and the receptor. So we have notch, we have delta ligand and the jagged receptor. So these are the two interacting partners in the notch signaling. And notch signaling is very important in forming the vulva of C. elegans, optic neurons, optic nerves of the optic nerve instruction, as well as the nervous system in mammals. So here is an example of vulva induction carried out by the notch signaling. The formation of anchor cell is mediated by Lin12. This gene is a homologue for the notch gene in C. elegans. So Lin12 is an important factor involved in vulva formation in C. elegans. As from the earlier classes, we already know Lin3 is a EGF-like protein which is secreted by the anchor cell. While what role does Lin12 play? In wild-type C. elegans, hermaphrodites, two adjacent cells Z1 PPP and Z4 AAA have the potential to become the anchor cell. So something predecessing the anchor cell. So many cells have the potential to become the anchor cell, but what does this Lin12 does? They interact in a manner that causes one of them to become the anchor cell while the other one becomes the precursor of the uterine tissue. So there are two cells which can become an anchor cell, but both of them signal via the Lin12 notch signaling mechanism, and one of them will become the anchor cell while the other becomes the precursor of uterine tissue. This notch delta interaction mediated mechanism of restricting adjacent cell fates is called lateral inhibition. So it is inhibiting the other cell laterally. So whatever cells that are present adjacent to each other in the lateral direction, it is inhibiting the formation of anchor cell and promoting only one cell to become the anchor cell through the Lin12 mediated mechanism. So Lin12, which is a notch ligand through the notch signaling pathway, is very important in vulval induction in C. elegans as well as the nervous system formation and optic neuron induction. So the correct answer to this question, as Ajay answered, is notch signaling is an example where the ligands and the receptors cannot be from far off cells. They have to be adjacent or in nearby vicinity. That is an example of juxtacrine interaction signaling. Moving on to the next question, question number 6. Which of these are false with respect to the ECM? Which of these is not a property of ECM? Statement 1. ECM components help in concentrating ligands near the receptor. Statement 2. ECM helps in instructive induction. Statement 3. ECM components have high affinity for ligands of many signaling pathways. And statement 4. ECM guides cell migration. There are 4 statements as functions of ECM and properties of ECM. You have to identify which of these is a false statement. This might be a bit tricky, but I think you guys can get to the answer. ECM components help in concentrating ligands. Meanwhile, Ajay has a question. In C. elegans, more than 10% cell die. Is it true for mammals also? You try to find out the answer for number 6 and I look for the answer for your answer. Option 4. Okay. Which of these are false with respect to the ECM? Ajay says option 4. And to answer your previous question, Ajay, in C. elegans, more than 10% cells die. Is it true for mammals also? For humans, you see, on an average, an adult human being, 50 to 70 billion cells die in an adult human being via apoptosis, via programmed cell death. So there can be many inducers of apoptosis, and it regularly uh, undergoes in our human body. Apoptosis is a counterbalance to the cell proliferation. So in each hour, in each day, 24 hours, our cell can divide. Similarly, apoptosis can maintain the balance, maintain the check between overproliferation. So we have apoptosis undergoing many through many pathways. One of such important pathways which I want to tell you about is the immunological pathway. There are certain cells called NK cells or natural killer cells which can induce apoptosis in their target cell. So suppose they find a cell in distress or with its machinery gone down, these NK cells can recognize these cells via their receptors presenting the MHC molecules, and then these NK cells have a type of ligand expressed on their cell membrane called Fas ligand. Fas ligand Fas ligand can induce apoptosis in its target cell. So this is one such example. Even RBCs undergo apoptosis and many other cells undergo apoptosis, and during development the number is still huge. So in C. elegans, if it's 10%, so it's more in mammals. And for the answer for to question number 6, he has changed you have changed your option to option 3. ECM components have high affinity for ligands of many signaling pathways. Okay, let me tell you what is the correct answer to this question. ECM helps in instructive induction is the correct answer to this question, which is false with respect to the ECM. ECM helps in permissive induction, not instructive induction, because ECM is not directly involved in signaling via it, but it produces a conducive environment for the cell to attain its fate. While all other components you see, ECM components help in concentrating ligands near the receptor is true. They have high affinity for ligands of many signaling pathways is also true. ECM guides cell migration. Cell migration is an important issue. Yeah, Ajay. I am here to clear your doubts. So, instructive induction, as we see here, let me tell you, instructive induction, we mean here that a signaling molecule is directly involved. Now you might argue there can be signaling molecules from the ECM which are involved. So there are certain examples, but which is not majorly the role of ECM. So if you see, there are certain integrin interaction which can signal through the ECM to the cell to differentiate into a certain thing. But ECM is the extracellular environment of the cell, and interactions with the ECM can create a permissive induction. What happens in permissive induction? It produces a conducive environment for the cells rather than being involved directly in instructing cells for a specific fate. So a cell in cellular state 1, changing plasticity of a stationary cell, remodeling of the ECM, then it can attain a cellular state 2. So just by remodeling the ECM, when we change the environment of the cell, immediate cell, the cell can attain a different morphology, not via direct instruction from the ECM, but because now its environment is restricting it to a certain fate. That's why, let me tell you, the stem cells that reside in our body, stem cells have a specific kind of ECM and an environment in which they reside called the niches or the stem cell niches. So when a stem cell remains a stem cell, it remains a stem cell such only in its niche. When it moves out of the niche and moves into a different extracellular matrix or an environment, it can attain restricted fate and differentiated morphology. That's why the bone marrow is the niche for hematopoietic stem cells. These stem cells can stay as stem cells in the matrix. Also, migration, ECM members can cause migration of the cells from one kind of ECM to another kind of ECM, migration to a new ECM causing change in cellular state, and transitional matrix enabling plasticity. So there is a ECM there in certain examples. There are ECM for transition states like epithelial to mesenchymal transition, which can transform one kind of cell to the other kind of cell. So ECM does not directly directs the cell to become a specific fate, cannot interchange its direction to a particular cell. Let's say a stem cell to become a blood cell, the ECM is not directly responsible, but the environment it provides from the outside is permissive induction. So that is the answer for this question. ECM is involved in permissive induction rather than instructive induction. So if you see instructive induction, it can be any signaling molecule. So let's say Wnt signaling is involved in instructive induction, or another cell, anchor cell, is involved in instructive induction. Anchor cell induces the fate of the underlying cells to induce vulva formation. So anchor cell will secrete something which will direct the recipient cells to form a vulva, while ECM is the surrounding environment and the matrix which will permit or permissive induction is the major part of the ECM. Now, on the other functions of the ECM helps in delivery of paracrine ligands such as FGF and Wnt, and proteoglycans are components of ECM which have affinity for many ligands such as Wnt. So Wnt has affinity for proteoglycans. So ECMs rich in proteoglycans can activate Wnt signaling pathway because the ligands can be concentrated near the receptors. They concentrate ligands near the plasma membrane bound receptors. That is what I just told you. So there are different kinds of migration that ECM can mediate. So there are different kinds of migration that ECM can mediate. Amoeboid, mesenchymal, collective cell invasion. So these are different kinds of migrations where we have degradation of the ECM, change in ECM properties, and there are certain integrins and attachment adhesion proteins which with the help of a ECM components attach to them and guide in motion. So ECM is involved in migration. ECM can concentrate ligands near the receptors. ECM have affinity for many ligands, and ECM also plays a role in permissive induction. I hope my voice is now clear. Maybe in between it breaks a bit, but yeah, thanks. Okay, let's move on to the next question, number 7. If epithelial mesenchymal transition causes cancers to metastasize, would it be a good idea to create an embryo that cannot undergo EMT? So this is a hypothetical experimental question. You know that EMT or epithelial mesenchymal transition is an important phenomenon which causes metastasis in cancers, which is a very deadly property of cancers. Cancer in one region can invade another region and start a new cancer. So, would it be a good idea to create an embryo that cannot undergo EMT? Now, there are four statements. Yes, EMT has no critical functions. So, okay, we can create an embryo such that there is no EMT because EMT has no critical functions. Statement 2, no, EMT plays a critical role in mesoderm and neural crest formation. No, because there is no way of preventing EMT. We cannot do it because we cannot prevent EMT. There is no such way. And yes, EMT can be easily prevented. Now, which of these statements do you think is the correct answer? Why would it be a good idea to create an embryo that cannot undergo EMT? I see Ajay has already answered. Option 2, no, EMT plays a critical role in mesoderm and neural crest formation. Okay, let us see what is the correct answer. Ajay has answered very correctly. EMT, it would not be a good idea to create an embryo that cannot undergo EMT because EMT plays a crucial role in mesoderm and neural crest formation. So let us have a quick look about neural crest and mesoderm. So the neural crest cells are certain type of stem cells which can give rise to many other structures such as important structures. So the peripheral nervous system comes from the neural crest cells. The cranial nerves, the cranial features, head, nose, and jaw muscles all come from neural crest cells. Now these neural crest cells are undergo epithelial to mesenchymal epithelial to mesenchymal transition because these neural crest cells are where originally the neuroepithelial cells which were lining the neural tube. Now at the tip of the neural tube where the neurulation happens, these cells undergo epithelial to mesenchymal transition. They move out from the neural tube and populate various areas. Neural crest cells are also responsible for melanocyte production, for peripheral nervous system, for cranial nerves and facial features. So EMT is necessary process which is required for proper neural tube closure and migration of neural crest cells. Similarly, the mesoderm tissue, mesoderm is an important tissue, part of the three germ layers, mesoderm, which forms muscles and kidneys and many other organs. So mesoderm tissue also undergoes epithelial mesenchymal transition from the epiblast in between the hypoplast to form the mesenchyme forming the mesoderm. So if you do not have EMT or we block EMT, then we may not have neural crest cells or mesenchyme forming mesoderm. That's why blocking EMT is not a good idea. Also to the other question whether at all we can block EMT or not. So EMT can be prevented and such strategies are in use for cancer treatments and therapies. So EMT can be prevented, but in an embryo where EMT is very necessary for formation of structures like neural crest cells and mesoderm, preventing EMT or abolishing EMT is not a very good idea. But to treat cancers in adults in extreme stages, blockers or small molecule inhibitors inhibiting EMT have been used, and such strategies are used for therapies in cancers. Blocking upstream signaling pathways such as TGF beta, NF kappa B. So these are the signaling pathway which trigger the initial stages of EMT where we have loss of contacts between the cell and loss of junctions. Targeting multiple EMT transcription factors such as snail twist. These are transcription factors which which promote more mesenchymal type. So such strategies are already being used in cancer treatment and therapies. So yes, it is possible to inhibit EMT, but inhibiting EMT or creating an embryo devoid of EMT is not a feasible idea because the embryo needs it for such important process like neural crest formation and mesoderm induction. I hope it is clear to the students. Yes, good. Let's move on to the next question, number 8. Now we have a match the following question. On the left hand side we have A, B, and on the right hand side 1, 2, 3, 4. Now you have to match the correct options. Specification and determination. On the right hand side we have fate is irreversible, capable of autonomous development in a neutral environment. Fate can be reversed by providing an opposite environment, capable of autonomous development even in a non-neutral environment. Now, which option do you think is the correct answer to this question? You might have to take a minute to consider this carefully. Specification and determination. There are many options, I think 6 options. You can choose between them. Specification and determination, whether fate is irreversible or fate can be reversed by providing an opposite environment, whether it is capable of autonomous development in a neutral environment or even in a non-neutral environment. Okay, option 4, Ajay has answered. Option 4 says A to 2 and 3. Specification, capable of autonomous development in a neutral environment. Fate can be reversed by providing an opposite environment. And B, 1 and 4. Fate is irreversible, capable of autonomous development even in a non-neutral environment. Okay, let us check what is the correct answer. Bang on, and Ajay is correct again. Option 4 is the correct answer. Very good, very good. So let's learn about specification and determination. So if I tell you in a short brief style, there are two different phases of cell commitment. When a cell gets committed to a particular fate, there are two different phases. Specification, cells begin to go toward a differentiated cell type, but this is reversible. Cells are able to differentiate into that cell type if cultured in vitro and away from other cells. So what it says is specification is reversible when cells begin to go toward a differentiated cell type. But what happens in determination, which comes as the next phase of specification, cells are further toward differentiated but are not functioning as the final cell type. Means it has reached an intermediate where it cannot come back again, but it has not even reached the final cell type. But this process is irreversible. Once a cell is determined, if move cells to new area of embryo, they will develop as if in original location. Now that the cell is determined to become something, now that a cell has determined to become a blood cell, you move it to any new environment. It will still form what it has to form. That's why we have in determination capable of autonomous development even in a non-neutral environment. Where we do not have a neutral environment, where the environment is actually permissive for something else, but since the cell is predetermined, cell has already reached the determination stage, it is still capable of autonomous development, and fate is irreversible in case of determination. While in specification, where the cell just has started uh, towards committed to a differentiated cell type, this process is reversible. cells Cell can develop autonomically away when it is kept in a neutral environment away from other cells. So which is answered correctly by Ajay. So here is an example, is a diagram uh, showing clearly, which will help you clearly understand the difference between specification and determination. So we have a fate map and differentiated tissue. In normal fate, we have two regions, region A and region B, which form these type of cells. The region A will form hexagonal cells, region B will form these square type cells or like a brick. Now region B is not determined. When B is transplanted to A, when some of the tissue is transplanted to A, we can still see red colored here marks the B region. We can still see hexagonal shapes. It means that the cells are not specified and in a conducive environment, in a permissive environment, because this is a non-neutral environment where A cells or region A is there, the cells will become A-like. Even if it comes from B, it can still become a A-like cell or a hexagonal shaped cell. Now after certain time, when region B is determined, now if you do the transplant, the cells will follow the brick type or square type morphology even in a non-neutral environment because they are already determined to become the region B type of cells. Now if region B is specified and you do not transplant it in a non-neutral environment, you transplant it in the neutral environment, it will still form the brick-like structures rather than the hexagonal structures. So that is the power of determination. But in specification, it is reversible. In determination, it is irreversible. I hope this is able to make things clearer and easy to understand for the students. Great. Now we quickly move on to the ninth question. Which of the following is false? So there are a lot of statements here. In embryos in which developmental fate is specified by autonomous mode, cells can independently develop irrespective of the neighboring cells. Shabri's experiment validates the autonomous specification model of cell differentiation. Driesch observed autonomous specification in sea urchins. Driesch's research shows that developmental potential of a cell is much greater than what it eventually develops into, and that neighboring cells play a critical role in preventing them from forming all cell types. And the fifth statement, Driesch's observation validates the theory of genomic equivalence. And Driesch's experiment shows that irrespective of the location of a cell in the embryo, it will differentiate into a particular cell type. Now, which of these statements do you think are false statements? There can be multiple, so you have to think about it clearly. Option 2. Option 2 is what Ajay is answering. Chabri's experiment validates autonomous specification model of cell differentiation. Okay. Any other statement do you think is false? Something about Driesch's observation and experiment? So a big hint is given itself in the statements. Driesch worked on sea urchins while Chabri worked on tunicates. Now if you remember some...
5 6 are correct, 4 is correct. I don't know about third, but 4 5 6 are correct. Well, I think all statements are.
Okay, let me now reveal the correct answer. Oh. Driesch observed autonomous specification in sea urchins. I think I marked the answer wrongly, maybe. Is it? Chabri validates autonomous specification is correct. Let me just check my answer again.
So just give the question. Let me try again.
I think I have marked the answer wrongly because Driesch observed autonomous specification in sea urchins is correct because each embryo, each blastomere when separated did form a full organism in sea urchin.
Only last option is false.
Yes, last option is
Developmental
false,
experiment.
but I think I have wrongly marked the third option. Let me just remove this. Yes, this will be the correct answer.
Because of this, the fellow went to become a philosopher and say,
Driesch's experiment shows that irrespective of the location of cell in the embryo, it will differentiate into a particular cell type. This is the false statement. Whereas Driesch's experiment proves the contrary. It shows us that location of the cell in the embryo is very important because following the sea urchin experiment, he also he also performed the glass experiment where he pressed the sea urchin embryo in.
And then he saw that there is a change in particular cell type that the embryo is forming. So let us see about these two experiments that the options are talking about. Chabri's experiment conducted in tunicates, where we can see here, which is a remarkable experiment for autonomous specification. What is autonomous specification? Autonomous specification occurs when a cell can independently follow a predetermined developmental pathway based on its inherent genetic and molecular makeup. So whatever is predetermined
Thing irrespective of its environment, or and that is called autonomous specification. So here in Chabri's experiment, what he did in the 8 cell stage of tunicates, the we already know that which blastomere is going to form what. So the A4.2 is going to form nervous system and ectoderm. B4.2, capital B 4.1 will form the mesenchyme and the muscle, while A4.1 will form the endoderm and the notochord. Now, when he separated the blastomeres and then cultured them, he could see that A4.2 still formed the ectoderm, B4.2 formed the ectoderm, while A4.1 formed the endoderm and the notochord, and B4.1 formed the mesenchyme and the muscle. The same exact way they were programmed in the early blastula stage. So even when on separation of these blastomeres, we see the same fate. That means this is an example of autonomous specification in which each blastomere is already specified what structure it is going to make. This was Chabri's experiment. Now this scientist Driesch in 1892 performed experiments with sea urchin where he separated the four-celled stage of sea urchin into four different blastomeres, and each blastomere could completely grow into a fully functional adult. So the major thing we can conclude from here is each blastomere is totipotent, contains all the genetic information required for to form an entire embryo with all its component tissues. And later this scientist also performed the glass pressure experiment in which he pressed the 8-celled sea urchin blastomere from different sides, and which caused the rearrangement of different blastomeres, and then he could see that still it is forming. The location of the cell is very important for the embryo's development, and whatever the previous location of the cell was and whatever program it contained developed autonomously to form the similar kind of structure when even when its location was changed. So few important conclusions from Driesch's experiment. Developmental potential of a blastomere is greater than what it develops into as a part of embryo. Each blastomere is totipotent. Developmental potential is larger than what it generally develops. So when you put all the blastomeres together, you do not get four different individuals. You get only one individual. That means each blastomere has the potential to form an individual. In normal development, neighboring cells prevent it from generating all the cell types a given blastomere is capable of. Here we see that when we have all the four together, we do not produce four organisms because the neighboring cells prevent it from generating the cell types a given blastomere is capable of. And the fate of a cell solely depends on its location in the embryo. So wherever it is located, the fate is autonomously certified and it can form the same cell fate irrespective of its position. That's why only the last statement, that the fate of cell is not dependent on the location of the embryo, is false, and all other statements are true. Moving on to the similar true false statement question number 10. Which of these statement is false? Autonomous specification leads to mosaic development. Conditional specification leads to regulative development. Syncytial specification shows that developmental potential of a nucleus depends on the concentration gradient it is exposed to. In Nuslin-Volhard's experiment, when the cytoplasm from the anterior portion of a wild-type egg is transferred into the thorax portion of the bicoid mutant egg, wild-type phenotype was
Roos conclusion of autonomous specification
developed from the remaining half due to conditional specification. Now, which of these statements do you think is false? Hmm. 4 and
is the correct answer.
option number 4 is correct answer. In Nuslin-Volhard's experiment, where the anterior portion of wild-type egg containing the bicoid transferred into the thorax portion of bicoid mutant, wild-type phenotype was not observed, whereas in the thorax region, head formation was observed. While in the fifth statement, it is also a correct answer, which I think I haven't marked it. Now that I've noticed it, I'll just mark it right now. Yeah. So Rue's conclusion of autonomous specification was incorrect. Everything of the statement is fine, but the complete organism would have developed from the remaining half due to autonomous specification. Since the dead half was joined with still in intact with the uh, alive half of Rue's experiment, so that was not allowing it to develop into a full organism because of conditional specification. In conditional specification, we have our environments and interactions of the cells determining the potential of the individual growth.
You already have that the other half of the embryo is going to form the full organism. That's why this statement is also false. So in autonomous specification, removal of a blastomere from an embryo causes the absence in the embryo of those tissues formed by that blastomere. This mechanism of specification produces mosaic pattern of development. So you remove one such blastomere in the 8 cell stage of sea urchin, then you do not see structures corresponding to that blastomere, while all other blastomeres form
Very correctly and independently of each other. That's why we have mosaic development. So we say autonomous specification is a pattern of mosaic development. But whereas in conditional specification, removal of a blastomere from the embryo can be compensated by other cells changing their fates. So what will happen in conditional specification? When you remove a blastomere from the embryo, it can be compensated by other cells changing their fates. Each cell can potentially give rise to more cell types than it normally does. This produces a regulative pattern of development wherein cell fates are determined relatively late. So cell fates are very lately determined in a regulative pattern. If one such cell is missing, then the other cell can partake its role and undergo conditional specification. And in syncytial specification, syncytium is a condition in which we have multiple nuclei in a single cytoplasm. Syncytial specification depends on morphogenetic gradients, which we see of bicoid nanos and all other anterior posterior polarity mRNA and proteins in the Drosophila syncytium. So here is a Nussnil-Volhard's experiment in a bicoid mutant. when you add to the anterior end of mutant, add bicoid mRNA or the anterior portion of the wild type, we have normal development of the structure, where we have acron, head, thorax, abdomen, and telson. Whereas if you add bicoid mRNA to the middle of the bicoid mutant embryo, we have head formation wherever we the bicoid around the anterior portion transplant. So that means the anterior head forming region of the wild type has some signal which is bicoid mRNA and protein, which causes head induction wherever it is injected into the embryo. So this is the case that we will have, which was not mentioned correctly in the statement 4. Whereas in wild type, if you add a if you add to the posterior of a wild type embryo, we have the anterior transplant. We'll have two heads and abdomen in the middle. This was an Nussnils Bollard's experiment, and Roux in 1890 showed that in two cell stage, when he ablated one half of the zygote with a hot needle, the dead tissue lying, living tissue. So the one half of the cell is destroyed and the other half develops fine to subtle stage of the neurula. Roux concluded that only half of the genetic information was partitioned to each of the two blastomeres. So two blastomeres frog embryo destroyed one blastomere with hot needle. Only one half of the embryo developed. So the embryo that developed from the egg was only one half. Since it was autonomically specified, it formed the half of the body, and since the other half was dead, it was sending in conditionally specified cues, which was not letting it develop to the whole full live embryo. So these two statements are therefore not the correct statement or the false statements. And with this, we have come to the end of this practice problem session. And Ajay had a question. In exam, how many questions are we supposed to be answered in 3 hours? Ajay, I might not be the person best person to answer this, because I really don't know in exams what kind of how many questions you will get. But I think the pattern will be similar. You will get MCQ type of questions, but that depends on the instructor. As a TA, I do not have much idea. Yes, Ajay, you wanted to speak something. You can tell me.
Sir, solving one question here, it takes too much time. And we know it is not going to get us a mark. Still, we answer it correctly or incorrectly. But in exam, we have to think twice before putting the option.
Yes, yes.
So many options, 6, 7, 8 options, and out of that, you have to find out one false.
Yeah. That really depends on the instructor actually.
The question from last examination, they are too much difficult. We are getting 100 questions in 3 hours, 180 minutes. Hardly 1.
No, I don't think you will get that much of questions. The maximum, see, in quizzes you get 10 questions and you have a whole week to submit that. So in exams for 3 hours, I don't think you will get 100 questions. But exact number I don't know. I just cannot say, just cannot tell you. Actually, I do not know.
So these are the questions from last exam or the first time there is exam?
These are actually quiz questions. These are not the final exam questions. So the quiz assignment that you have, it is similar to those. So I'll stop presenting.
Usually in multiple choice question there is only one correct option.
Yeah. I know.
This time, first time I have to see whether there are other correct options or not.
Yeah. It might be a little tricky, but this is how the course is, and the instructor wants us to conduct the exam, or the NPTEL is conducting exam, and you have to go through it. So it might be really little difficult for you guys, but I think your concepts are very clear, and you will be able to do really good in the exams. So you do not have to worry about that.
Thank you sir for your guidance and motivation.
Yeah, yeah, yeah.
I hope you
So
can close the sir. Please do something with the mic. At the last thing, it was
yeah, I know.
when I
I know.
started
I think for
that
today it is creating
I
some
turn on the
problems.
capture. So whatever you are telling, I am seeing there as a editor. I reduce that my sound. Whatever you are telling me, I can read it. Please do
Okay,
that.
yeah, I apologize
Sorry
for
to
today, but
interrupt
I
you.
think it's some technical problem specific to today because I didn't face any difficulty in the other classes. Let's hope that in the next class it gets corrected, and do not worry, you will have access to the YouTube recordings. Let us see whether my voice breaks there or not. But you feel free to ask any doubts or stop me anytime when you want. And with this, I'll close today's session and meet you
Sir,
guys.
one more thing. The
Yes, yes, yes.
Parth sir is not coming on. Not successive 2 weeks. They had not appeared. Classes are being there posted in emails, but it's not coming. Any problem with him? He's all right or something?
Oh,
Video call.
actually, yeah, I have no contact with Parth also, but I don't know. I think he should attend his. If there are lectures, I think he must be taking on the scheduled time. Where you
So, successively last 2 weeks, because I always, what you say, I'm very particular about my time.
yeah, yeah, yeah.
As soon as 8 p.m. I come, it's not nobody
Yeah,
there. That's why I get better
yeah,
time.
I am also not aware of how Path is operating because he is in a
I
total
have
different
no way to
institute.
communicate or ask him apart from email. But if I write something on the email, it will go against him. That's why I'm asking.
Yeah, yeah,
He's
yeah.
all right or not? Just if he's okay,
I
no
think he's all
problem.
right, but maybe he was not able to make it at the particular time that he was allotted to, or he's recording his lectures and putting it on YouTube. Maybe he was busy for some time, but for me as well,
I,
I am sincerely like uh, putting on my classes, preparing for it, even when I have less students or no students. So it doesn't put me down. What I have to do is for the betterment of the students, and as a TA, since this session is recorded, I have to put forth my questions and
I understand
put
it
the
takes too
answers.
much time
Yeah,
to prepare 1 hour or 2 hours class,
yeah, yeah, yeah.
and if there is no audience, it's very bad.
It's
That's
okay.
why
It's
I asked
okay,
him.
Ajay. If
Saturday time is too much odd.
yeah, yeah, I know, but this is the only time that as a PhD student I can afford freely for my classes. And this was the time for the earlier NPTEL classes also. I used to take other NPTEL courses which was not from the domain of biology, but so far so good.
Okay, sir.
Yes, Ajay,
Yes, Ajay,
I think I
I
lost
think
my
I lost.
connection. Oops.
Okay, sir. Bye.
Yes, thank you,
Yes,
Ajay.
thank you,
I'll
Ajay.
see you in the next
See you
week.
in the next.
I'll end this call
I'll
now.
end this call now.